Good morning, students. Today we'll clear the concept of antigen. Antigen is a substance, usually a protein in nature, but it can be polysaccharide, it can be lipid. It generates specific immune response and induces the formation of specific antibody or specially sensitized T cells or both. Although all antigens are recognized by specific lymphocytes or by antibodies, only some antigens are capable of activating lymphocytes. So molecules that stimulates the immune responses are called immunogen. Epitope is immunologically active region of the antigen or immunogen that binds to antigen specific membrane receptors on the lymphocytes or to secreted antibodies. These are also known as antigen determinants. In this figure, you see this is a bacteria. It is having different types of antigenic determinants. These are known as the epitopes. These are the epitopes where the antibodies, they can Y-shaped structure antibodies, they can bind with it. So epitope or antigen determinants, they are the structures which are present. So there is a difference between antigen and immunogen. Antigens are the foreign substances which binds with the product of immune responses, that is antibodies or the T cells, whereas immunogens are the foreign substances which have ability to induce the production of immune response products like antibodies, activated T cells. So all the antigens, they are not immunogens, but all the immunogens are antigens. Autoantigens. Autoantigens, for example, person's own self antigen. For example, person's DNA, corneal tissue, thyroglobulin. Alloantigens. They are antigens which are found in different members of same species. For example, RBC antigen. For example, blood group A have different antigen, B has different, AB has different, O has different. These are known as alloantigens. Then is heterophile antigens. They are identical antigens found in the cell of different species. For example, cross-reacting microbial antigens. Then is adjuvants. They are substances which are non-immunogenic alone but enhance the immunogenicity of any added immunogen. Then the chemical nature of the antigen. So as I've told you, the antigens, they are basically the proteins. So we can say that maximum they are the proteins, pure proteins or glycoproteins or lipoproteins. So they are very good immunogens. Second is polysaccharides. Pure polysaccharides and lipopolysaccharides are good immunogens. Then is nucleic acids. Nucleic acids are poorly immunogenic. However, they may become immunogenic when single-stranded or when complexed with the proteins. Then is lipids. In general, lipids are non-immunogenic, although they may be haptens. That is, when they combined with any protein, they become immunogenic. Then two types of antigen on the basis of order of their classes, that is exogenous antigen, endogenous antigen and autoantigen. Exogenous antigen, as you know, these antigens, they'll enter into the antigen presenting cell, they are processed in the antigen presenting cell, and the processed peptide come to outside as MHC molecule. Examples are bacteria, viruses and fungi. Endogenous antigens, they are body's own cells or subfragments or compounds or antigenic products that which are produced inside the cell, right? And then they are processed and their peptide, they will come out on the body on the surface of the cell as MHC component. So their example is blood group antigens, HLA, etc. Then is autoantigens. The autoantigens are usually a normal protein or complex of proteins, sometimes DNA or RNA, that is recognized by the immune system of patient suffering from specific autoimmune diseases. So autoantigens, they are person's own proteins which becomes antigenic. On the basis of immune response, we have got two types of antigen. One is complete antigens or immunogen. Second is incomplete antigens or haptanes. Complete antigens means they are basically the proteins having the high molecular weight, more than, very important, 10,000 daltons. So they can induce the immune response at their own. Whereas incomplete antigens or haptanes, they are foreign substances which are usually non-proteinaceous substances. Their molecular weight is very less than 10,000 daltons. They, when combined with the carrier molecule, which is a protein, forms haptane carrier complex, which becomes immunogenic and it provokes the immune responses. Then the antigens, basically they are of two types depending upon their dependence on the T cells, that is T cell dependent antigen and T cell independent antigen. T cell dependent antigen means they require T cell, especially T helper cells, to induce the immune response. For example, all the soluble protein, right? Then is T cell independent antigen. These cells, they don't require the T cells to induce the immune response. On this basis, basically they are of two types: T cell independent antigen 1, T cell independent antigen 2. T cell independent antigen 1 means it does not require T cell to induce the immune response. For example, lipopolysaccharide means they don't require it. Second is they don't require the T cells, but they require cytokines which are produced by the T cell. Means indirectly they require T cell, but not directly they are requiring the T cells. For example, cell wall and polysaccharide. So in this diagram, you can say that we have got two figures. One is T cell independent. First one is T cell independent antigen. That is the B cell is there, right? It is having the immunoglobulins, the antigen, it binds with it and it induces the immune response and ultimately huge number of antibodies they are produced. Second, in this case, B cell is there, immunoglobulin is there, antigen it binds with it, but it is unable to induce the antibodies. Then what happens? It will process the antigen and the processed antigen comes in the form of MHC. That MHC binds with T helper cell and T helper cell then produces the cytokines and ultimately stimulates the B cell to produce huge amount of plasma cell and memory cell to produce the antibodies. So this is T cell dependent antigen. Next term is the super antigen. These antigen activate T cells non-specifically, means they are not specific. They can bind with any T helper T cells. So they are of two types, exogenous and endogenous super antigens. Now what happens in the case of exogenous super antigens? They are capable of activating T cell APC complex exogenously. In this figure, first figure, you will see that this blue colored structure, that is exogenous super antigen, it is not processed inside the antigen presenting cell. It comes from outside, bind with this complex, that is T helper cell APC complex, and as it firmly binds with this, it stimulates the production of huge number of cytokines and leads to the immune response. These antigens, they are never processed, very important. Example is toxic shock syndrome toxin produced from the Staphylococcus. Second is endogenous super antigens. They are endogenous because they are processed and present into APC cells in irregular manner. They are not presented through the MHC molecule. This is very important. Neither exogenous nor endogenous, they are not presented through the MHC molecule. They are presented outside the MHC molecule. See here, it is present here. From here it will come out, and ultimately it will stabilize this complex of T helper cell and APC and leads to production of huge amount of cytokines. Example is mouse memory tumor viral antigens. So very important is the diseases associated with the exposure of superantigens are due to hyperactivation of immune system and subsequently release of biological active cytokines by the activated T cells. Very important, they are non-specifically attached. These antigens, they are attached outside the complex and they are not associated with MHC in any form. Common antigen, examples of antigen: animal or plant proteins, bacteria, viruses, chemicals, dust, various drugs such as antibiotics, foods, milk, chocolate, for example wheat, perfumes, plants, pollens, as well as smoke. Next is the epitope. As you know, antigen binds with the B cell or T cell, right? Now, the smallest part of antigen or immunogen that can be recognized by B cell receptor or T cell receptor is called epitope. So, epitope is the smallest part of antigen which is recognized by the BCR or TCR, right? Now, these two, they are different. B cell, as you know, B cell can bind with the antigen which is present on any organism directly through the B cell receptor. That is, B cell, it is having B cell receptor, and this B cell receptor directly without any processing can bind with the epitope. So B cell epitope is recognized by BCR, B cell receptor, and it can be free soluble molecule or degraded molecule or fragment or surface bound molecule. So such epitopes, they can be polypeptide, can be lipid, can be polysaccharide, can be organic compound, right? And their size is larger than the T cell epitope. Very important. B cell epitope can be linear, can be conformational. So in this figure you can say that these are the epitope. Linear means continuous stretch of strand is there, right? And conformational means that is, it shows 3D structure folding is there. That is, different folds, they will, when combined together, they will form the conformational epitopes. Now coming to the T cell epitope. T cell epitope is recognized by the TCR, that is T cell receptor, right? So TCR is present on the T cells. As you know, T cells, they are of two types, CD8 and CD4. So T cell epitope is always a linear peptide. It is always a peptide, very important, right? And secondly, MHC molecule is required to display this peptide. If MHC molecule is not there, T cell epitope will not be functional. So it is bind with the MHC molecule. The size of T cell epitope is different in CD8 and CD4 cells. In CD8 cells, it is formed by 8 to 12 amino acids, whereas in the case of CD4 cells, it is formed by 12 to 17 amino acid. And this epitope, T cell epitope, is always linear, right? So in this diagram, you can see that here in this B cell, B cell receptor is there, and antigen having the epitope directly binds with it. But in this case, the cell, it is having MHC molecule, and on the MHC molecule we have got the processed epitope, processed peptide, only peptide which can bind with the TCR. So In this, it is only a protein. Here it can be protein, it can be polypeptide, it can be polysaccharide, it can be lipid. Any form can be there. An antigen can directly bind with it, but here processing is required. So I hope the concept of antigen is clear to you.